All right, we're live. How's it going, everyone? All right,
Cool.
So we're at 8:30. Shall we get this show on the road? Hello, everyone. Welcome to today's session of the Escarpment Labs webinar series. Escarpment Labs is a yeast producer based in Guelph, Canada. We do work with breweries all over the world to solve fermentation problems and create new flavor opportunities. I'm Richard Priest, co-founder of Escarpment Labs, and I'll be the moderator for this webinar. If you're joining us live, please tell us who you are and what you've been doing the past week to pass the time. The Crowdcast platform is an excellent way to get conversations started, and community is something we need right now. Today, our topic is Yeast Basics Part 1, How Yeast Works. I'd like to introduce today's presenter. Today, we have Nate Ferguson, co-founder of Escarpment Laboratories and professor at the Niagara College Brewing Program. Nate graduated from the University of Guelph with a degree in biochemistry, and he's a treasure trove of knowledge in all things fermentation. For those joining us live, please feel free to start a discussion on the Crowdcast platform, ask questions and upvote questions. It's really great for keeping these conversations going. I'll do my best to monitor the discussion and follow up with our speaker if needed. For those that are tuning in after the live cast, thank you for checking out what we have to offer. A recorded version of this webinar will be available. If there are questions that we don't get to, what we're going to do is we're going to compile those into the Q&A session that we have planned for the end of this Yeast Basics series. So without further ado, I want to give Nate the floor to tell us all about Yeast Basics Part 1, How Yeast Works.
Awesome. So with that, we'll just get the everything kind of loaded up here. And there we go. So welcome to guys. Today we're going to go through kind of the basics of how fermentation works, or more so how yeast works. Now, there's going to be a handful of different goals for today, but the big goal we're going to get to is to get everyone to have a more holistic view of yeast. I don't mean holistic in a somewhat flippant or lighthearted way. I mean, I want you to treat it a bit differently than you probably do, or I want you to conceptualize it a bit different than you probably already do. We're going to get more into this, but by the end of today, you're going to have a better idea as to how yeast actually functions as an organism, not simply as a fermentation bean that we use in the brew house. Understanding how the organism actually functions will allow you to much better understand and appreciate and alter your actions in the brew house, and that help you understand why yeast are doing the certain thing they're doing, and how you can help manipulate them to do the things that you want them to do. So without further ado, we're going to get started. Now, before we get really into it, I just want to give a kind of shout out to the rest of the team. Escarpment Labs currently is 16 staff strong. Two of the staff here aren't pictured, but this is the entire team. We're all trying to get through all this stuff together. We're still currently open, and we're I just want to say shout out to all of those guys and say thanks. If it wasn't for them, none of this would have happened. So to kind of structure how the next 3 lectures are going to go. So today, March 23rd, we're going to go over what yeast is. We're going to talk about some fermentation basics, what it likes to eat, how it eats that, something that probably most people haven't really thought about, what it likes and what it doesn't like. And then finally, we're going to leave on the different types of yeast you're going to typically find inside of breweries. Now, the next session is on Thursday. We're going to go through what flavors yeast cells produce, how yeast cells grow, and all the different positive and negative flavors it can produce. And then finally, on March 30th, so this time next week, we're going to go through controlling flavor compound production. We'll look at fermentation temperature, pitching rate, yeast cropping, management. All those good things are going to have a big impact. Now, the goal of today and the goal of this entire 3 sessions is to instill the basics. So each one of these different subpoints you see on here could easily be, a 2-hour presentation that gives you all the nitty-gritty detail for it. The goal of this is to show us the basics and hopefully show you something you didn't know about yeast that you've maybe taken for granted. So a few disclaimers before we get going, and that's just kind of what I just mentioned. This is the basics. We're here to introduce these topics. Now, if there is something you want us to talk about further, please leave it in the comments. That's why we have the moderation. That's why we're using Crowdcast, so we can have these conversations going on the entire time. The goal here, though, I can't stress this enough, is basics. We're not trying to teach scientists. We're trying to teach brewers. There's a few other bits to this. We're going to be talking about how yeast works, but with the main focus of how you can become a better brewer by arming yourself with this knowledge. I am not guiding any of these things towards yeast researchers. If you want to be a scientist and a researcher, awesome. I would recommend it. It's wonderful. But the goal of this series really is for brewers. This means that we will be skipping over some portions of detail that I would like to go into, and we can go into it potentially at the end of the conversation or in the Q&A next Thursday. But we're going to be going over and focusing on the basics so we all have the same level of knowledge. And so for today, we're going to go through what yeast is, how it eats, what needs to be healthy and make good beer. It's an important bit. What makes it unhappy or stresses out or makes it angry. And finally, how humans and yeast are different. Now, to start this whole thing off, I can't think of a better way than this. Yeast, or Saccharomyces cerevisiae for the most part, should always be your #1 employee, always. Employee of the month in every single brewery should be a picture of a yeast cell. No questions asked. It is by far the most crucial ingredient in any sort of alcoholic fermentation, because without it, there is no alcohol. And without no alcohol, there really isn't a point to anything we're doing. Historically, or kind of in a modern context, that's a bit different. For the most part, it's true. Now, I have some issues in the way in which some people will conceptualize this topic. A lot of people want to jump straight into the deep end on this and make yeast fermentation as complicated as you possibly can. They want to jump into third-year microbiology and say, go start from there. We're not starting from there. We're starting from a much more basic, kind of simplistic viewpoint. Now, other things I see people do when it comes to yeast, I see a lot of people treat it like it's an ingredient. Now, in doing so, people often become very confused, and we answer emails with this all the time, where they added the yeast to an ingredient. They treat the yeast like an ingredient. They didn't understand the stresses and pressures that are put on the yeast cells, and they don't understand why a certain flavor compound was produced. I would recommend that you don't treat yeast just like an ingredient. I'd recommend you treat it more like a pet. And it is that way. It is a better way to talk about it. And many brewers do. The first brewery I ever worked with, everyone who worked there before I got there talked about the yeast like it was like the brewery's cat or the brewery's pet. Treating your yeast cells like they are a part of the team, I mean it literally, and that they are an animal as they are, something that is actually living, is a much better way to work with it. And in doing so, you'll understand what makes it healthy, what makes it sad, what makes it stressed out, what makes it unpredictable, what it needs, how each different yeast cell differs. If you have 4 dogs, they're all going to have their own little quirks and things that make them tick. That's the goal of this. I would recommend that you treat your yeast cells more like pets. Understand how each one of them works, understand how each one of them functions and what you need to do for each one to make them happy. You want to form a relationship with your yeast. You want to understand how it actually works. Otherwise, it's unpredictable. You're going to have bad beer and you don't know what's actually going on. Think of it like Honey, I Shrunk the Kids. If we're going to be delving into a handful of topics in the next coming weeks that are going to be a lot more complex than today. Believe me, a lot more complex than today. And it becomes very useful if you can help get yourself in the mind of a yeast, which sounds pretty nerdy and it sounds kind of esoteric in a sense, but it can be incredibly useful. If you can understand what stresses, or if you put a yeast cell in the following scenarios, is it going to be happy? Is it going to be stressed out? Is it going to be, is this a toxic environment? Is this a good environment? Is this yeast cell coming from a toxic environment or a healthy environment? Being able to kind of understand the state the yeast is in, although I know this sounds very fluffy, can be very, very useful, incredibly impactful when it comes to your beer quality. So, what I want us to do is take a little quick walk and compare and contrast kind of humans and yeast. So are we doing this throughout the entire presentation today? Because I want to showcase to everyone that we're not very different. When you start to understand the differences and, more important, the similarities, you start to realize that the way in which some people treat yeast as if it's this strict ingredient or just a yeast plus sugar equals ethanol, simplistic device like that, doesn't really quite work. Now, we're about to go into something that's a bit more in depth, but I need to remember here that we're only going to talk about things that are going to make you better brewers. So whenever I do bring up a scientific diagram or something like that, I'm doing so with the intent that it is going to make you a better brewer. I just need to remind everyone, because sometimes that can pull people away. So what we see here is a basic example of what a yeast cell is. Now, if you want to go into the cell biology and things like that, I'd recommend it. It's a lot of fun, but that's not the point of today. What I want to show you guys today is kind of the basic structure of it. We've probably all seen a yeast cell before, so I want to take a mental picture of this and then compare it to the human cell we have next to it. They're incredibly similar. they're near identical. They're very, very, very similar. Humans and yeast cells have an incredible amount of similarity. And I don't understand why people decide to then, based on this, treat humans and or pets or animals or multicellular life different than single cellular life. They're very, very different. They're incredibly different. Now, some people, and this is where we see people get kind of confused, is they try to treat yeast like it's not a living organism. I'm going to be hammering this in a handful of times. So if you get tired of hearing that, I'm sorry, but it's really important. Human cells are much more complex. So we form multicellular systems. We know the yeast cell is a much simpler version of that, but the basic structure of how it works is exactly the same. Now, there's a very common rule that you see across all sorts of different societies. It's called the golden rule. It's as simple as this. Treat others, and in this case, the planet, as you would like to be treated. Do unto others as you would want done unto yourself. If you add yeast onto this, a lot of the problems you will have inside the brewery will go away. If you know how to treat your yeast cells properly, healthily, to keep them happy, a lot of the problems you have will fade. And a lot of the problems brewers have is that they're doing things and not realizing that they're doing something that's really negative to their yeast cells, whether it's required or not required based on the brewery's outline. So what we'll be exploring is how we can actually treat our yeast cells healthy. Now, what a handful of you are probably thinking is, that sounds wonderful, do unto others as I would do unto myself, but I don't know how to do that to yeast. I don't know how to treat yeast properly. And that's what we're going to really focus on today, is that bit right there. How do we actually make yeast cells happy? What do we do to make them stressed out? And be able to understand that, because once we understand those principles, we can then jump into the metabolism, the cellular biology, get really nitty gritty and figure out how to make these things even better. So with that, we're going to kick it really off. So we're going to go through fermentation basics as to why it does it. Now, fermentation's always fun. I remember the first things I ever fermented was just simple sugar water because I was about 16 years old, and there's a story behind that, but now is not the time. Fermentation's fascinating. It's one of these things that has really pushed humans to arguably be everything we are today. There's a really cool theory about that humans essentially drank their way to civilization. That's how farming and agrarian cultures came about. There's something magical or very human, very not necessarily human, but very primal, so to speak, almost like fire about fermentation. It's fascinating. And humans have been trying to understand this for arguably as long as humans have existed. And we can make this very simple. We can break this down to its simplest bits, which is sugar plus yeast equals fermentation, which is alcohol and CO2. But there's a really important question that needs to be asked here, and that is, why do yeast cells do this? A lot of we take this for granted all the time, that when you add yeast cells to sugar, they're going to produce alcohol and CO2. But there has to be a reason behind it. There has to be, and that's one of the things we're going to tackle today. Now, the obvious answer to this is energy. We see here sugar plus yeast, which we see inside the cell here. We see fermentation. It generates ATP, an energy currency we see inside the yeast cells. The byproduct of this is ethanol plus CO2. But this misses over a huge amount of depth and a huge amount of reason behind it. Now, this is, we want to go a bit more in-depth into the entire process. This is what actually fermentation looks like from a metabolic standpoint. Glucose enters the cell. We generate two pyruvate molecules, get some energy out of it in the form of ATP. Pyruvate turns to acetaldehyde, releases all the wonderful bubbles that we see come out through our fermentation and carbonate our beverages, which then turns into acetaldehyde, and then finally into ethanol. We're going to go into this chart a lot more next week. Later on this week and next week. But we can break this really down into the metabolism, into the individual molecules. It's kind of fascinating. Now, the question I would then pose after kind of learning all this is, why does a yeast cell perform fermentation? And I know brewers who have been brewing for 20, 30 years who have never actually asked themselves the question, why yeast cells ferment? Why do they actually do this thing? It's a really odd thing for them to have actually occurred. And the going hypothesis for this is something like this. Yeast produced ethanol to prevent anything else from eating that food. So what I want to, give ourselves a little story here. Ethanol kills off almost all bacteria, molds, more importantly, things that are going to try and consume the food that yeast is trying to eat. Alcohol effectively is a survival mechanism. Yeast cells come down, they find some food. How they get there is varied. It could be insects, could be all sorts, could be mammals, all sorts of different things. But a yeast cell hits a sugary substance, usually during harvest and early fall, late summer, and it starts consuming all that sugar. Now, there are tons of organisms in our environment, things like E. coli, Salmonella, Staphylococcus, all sorts of bad things in the environment that also want that food. There's also molds, black molds, white molds, all sorts of stuff that really want that food as well. And the way in which we believe yeast cells kind of surpassed all these different organisms, outcompeted it, is by its ability to produce alcohol. Most organisms die in the presence of alcohol. Alcohol is a poison, and yeast cells actually don't like it all that much as well. They tolerate it, as is a better way to look at it. Humans, much like also many other complex organisms, we've learned to adapt to it. We can tolerate it, but it is poison, and that's part of the reason why we get hungover. Yeast cells produce this thing because it has survival benefits to them. It is the lesser of two evils for them, but they don't like it. And I think this is something that a lot of brewers miss. They think that yeast cells have no issue with handling alcohol, but they do. It's not a pleasant thing for them to be around. Just like for us, it's not a pleasant thing for us to be around. We enjoy it, but for different reasons. Now, here are the things. Do yeast cells want to ferment? And I mean this, like if you put yourself in the body of a yeast cell, do you want to ferment? And the answer pretty clearly is no. Yeast cells is not efficient. Sorry, fermentation is not an efficient way to produce energy. It's actually horrifically efficient. Aerobic respiration, which is what we do, humans. I hope you're a human. We require oxygen. That's why we have to breathe in. We have to respire. This produces 18 times the amount of energy that anaerobic respiration does, that fermentation does. It is a far more energy efficient system. And yeast prefer to do that, much like we do. Yeast prefer to be in this aerobic, high energy environment, just like us. Now, we prefer them to be anaerobic. We prefer them to produce alcohol, and they tolerate that. But overall, they want to be aerobic just like us. So when we deprive them of being anaerobic for long periods of time, be it say a fermentation, we are inducing a stress on them. There's no other way to state it. We are stressing them out by doing this. Yeast cells in their perfect environment would be at slightly above ambient temperature and a completely aerobic. That would be their ideal scenario. But we push them to be otherwise. So we are constantly stressing them out. Now, just to give you guys a reference point for all of this, the metabolism between humans and yeast cells is incredibly similar. And you can see this in this chart right here. Both humans and yeast cells both can go through aerobic respiration, which you see on the left-hand side of the chart. And as you can follow it through, one molecule of glucose comes in, produces 2 molecules of pyruvate. Pyruvate then gets broken down and turned into 36 molecules of ATP, or energy. ATP is the thing that powers the entire body, the entire cell. Now, if we go in the absence of oxygen, under fermentation, yeast cells will only produce 2 molecules of ATP, which you can tell is, based on basic math, 36 divided by 2 is 18 times less energy for the same amount of sugar in. Oxygen has a huge impact when it comes to producing energy in healthy yeast cells. Now, humans can also go through this. If you were exercising and your body is not able to get enough oxygen to your blood or to your muscles will start respiring under anaerobic conditions. And unlike yeast cells where they produce ethanol, our cells actually produce lactic acid. We might talk about that at a later point when we look at lactobacillus and things like that. So that's fermentation. That's kind of the Coles notes of it. We're going to into a lot more detail of that next week as to how it actually occurs and the stages of that. So if you're looking for a bit more detail, that's later on this week and next week. We're trying to get you guys the basics in today, just how yeast works from a bit more of a holistic standpoint. So we're now going to go into how yeast cells actually eat, because a lot of people don't think about how yeast cells actually consume their food. Now, yeast cells are a much simpler organism than humans. Now, this means that, by and large, they're unable to consume as much complex foods. Yeast don't have a stomach. Yeast don't have a small and large intestine. Yeast don't have these ability or these structures present in order that we do that allow us to break down these complex, very dense foods. Yeast cells need their food already pre-broken down. They can't break things down themselves to a variable degree. But the reason we consume and the reason they consume are very similar. They do this for 2 main reasons: to produce energy and to build and repair cells. Whether it's a human, whether it's a dog, a cat, an ox, or a yeast cell, they're all just trying to produce energy and build and repair cells. So we're going to look at this. Now, I want to remind you again, I'm only going to show you things that beneficial for you to become as a brewer. So when I show you the chart like this, I'm not trying to scare you. There actually is something beneficial here for you guys to learn. Now, we're going to cover almost all of this chart over the next 3 webinars. Well, this being the first one and the next 2 webinars. But if you take a quick look through this chart, you'll notice that almost all the standard fermentation profiles we see, or fermentation molecules we see here. Sugar comes into the cell, we get pyruvate, which then goes to acetaldehyde to ethanol. We can also turn to acetyl-CoA, TCA cycle. We can start doing all the different things that we see. We have diacetyl esters, fatty acids, fusel alcohols, all sorts of really higher alcohols, all sorts of things that we associate with fermentation. We're going to be looking at this chart a significant amount next week, or next lecture. But what I want to point out here is this, and that there are only 2 real inputs. Now, how we vary these inputs and how we can vary the health of the cell is going to cause a huge shift in the way in which the yeast cell responds. If I reduce or increase the amount of sugar amino acids, we're going to see flavor and cell health changes. If I alter the state the yeast cell's in and I give it the same food sources, we're going to see variances in it. There are very minimal inputs for yeast cells. They're not a huge amount. But how those are used and what state and what's kind of breakdown those sugars are in, or amino acids are in, is going to have a huge impact on the health and the flavor profiles of the cell. So we're going to start diving into these. So the basic inputs are sugar and amino acids. So we're going to go through the 2. Sugar is mainly simple sugars produced during the mash. This is why we need to mash our grains when we're trying to make beer. Now, we used to consume up to maltotriose, and maltotriose is what we call a trisaccharide. So it's glucose, glucose, glucose. 3 molecules of glucose in a row. Now, yeast cells will also consume maltose, which is 2 molecules of glucose, glucose, glucose. And it'll also consume glucose, just pure, simplest form of sugar you can possibly find. You'll also see different types of yeast cells. Almost all will consume sucrose. Fructose is also, almost consumed across the board. Some will consume things like raffinose or malabaiose, but that's a bit more specialized. All of these simple, easy to break down sugars are what yeast cells can consume. Now, they won't consume things like starches that we can consume. Like, you won't see a yeast cell chomping on a piece of rice. It's not going to happen. Yeast cells need super simple sugars in order for them to consume it. Now, the main precursors, all these different sugars they consume are the main precursors for ethanol. When we think about fermentation, sugar in, or in this case, up to maltotriose, ethanol out. Now, one thing that people often forget about this, though, is that whenever the yeast cells are growing, whenever they're budding, a large portion of that carbon, a large portion of that sugar that's consumed, much like when a human being is growing, it's not just going towards, say, CO2 production or, in this case, ethanol. It's actually going to help rebuild the cell and help repair the cell and grow the cell. Cells use sugar, which is made of carbon, for everything within the cell. It is one of the most fundamental building blocks for yeast cells production. Now, the other fundamental building block for the yeast cell's growth and maintenance are amino acids. And these are the building blocks for all proteins. And proteins are the things that allow and confer for all sorts of functionality, whether or not it's the yeast cell's ability to produce ethanol, whether the yeast cell's ability to stay, maintain itself and stay integral as it's in dormancy, or whether it's a human wanting to move their arms around. Proteins that allow us to do that. Now, the amino acids we see inside of the wort are mainly composed of FAN, which is free amino nitrogen, which essentially is small chunks of amino acids, ammonia, the purest form of nitrogen, small chain proteins or peptides, and other nitrogen sources. Nitrogen is the big key word here. The yeast cell needs nitrogen of bioavailable sources for it to do its thing. Now, almost all of these compounds are derived from malt. So if we're making proper 1040 or plus wort, we're going to be giving the yeast cells tons of food and tons of these precursors to work with. And other thing that's often not talked about is this amino acid, this FAN, is one of the main precursors for flavor compounds inside of a fermentation. Now, this is for good ones and bad ones. We'll talk about that on Thursday and next week. Now, if we want to look at how yeast cells actually consume, yeast cells don't have a mouth like a human. They don't have one of these. Yeast instead use transport proteins. Now, these are the proteins, as we talked about beforehand, protein the things that allow a yeast cell to do all the things. They're also the things that allow us to do all the things we want to do. Now, unlike us, where we have one consistent mouth to eat all the different things we need to consume, yeast has specialized transporters for every single nutrient it needs. So for sugars, they have things called hexose transporters. Sugar is another term for them is hexoses. So that's where the name comes from. We have FAN, which typically goes through a GAP transporter, and we have micronutrients, which typically go through specialized transporters or generalized transporters for that nutrient. Each nutrient has its own transporter, and the yeast cell regulates how much it has available, how much it has inside of it based on these transporters. And we're going to take a look at them quickly here. So what we see here are two different hexose transporters on the outside of the cell membrane. So we have one on the outside, we have some on the inside. This protein actually spans the entire cell membrane, and as sugars are present in the outside, this pump effectively will pump these sugars from the outside across the membrane and inside the cell so the yeast cell can use them. Same thing will happen for a whole slew of different transporters. We see this happen with gap proteins as well, where this can even be used between cells or from the outside of the cell to the inside. We have whatever we're looking for on the outside, and it gets pumped from one side to the next. It's as simple as that. Now, I want to take a quick second here, just kind of compare humans versus yeast cells, or yeast cells versus humans. I find this a useful exercise for us to go through when it comes to us actually understanding how humans and yeast cells are similar and different. So when we start talking about protein, when it comes to humans, you need a certain amount of protein inside of your diet. The equivalent that for yeast is that they need a certain amount of family amino acids in their diet. We need protein, they need amino acids. Simple as that. Now, when we need carbohydrates, things like breads and pastas, things like that, they need simple sugars. If I try to feed, say, bread, just straight up flour to a yeast cell, it's not going to do much. There's a tiny amount of simple sugars in flour like you get at the grocery store, very small amount, but not enough for it to really sustain it in the fermentation. And finally, if I'm looking for vitamins and nutrients, we both need effectively the same thing. The amount of the vitamins and nutrients that we need are very similar to vitamins and nutrients that yeast cells need. We get them from our food. Yeast cells should also get them from their food, being wort. Now, if we try to ferment things such as sugar to make just, say, hard seltzer, or if I'm trying to make mead that has very low nutrient and vitamin content, that can start causing problems. I haven't actually given it the proper nutrient it needs, much like if I don't have the proper nutrient and become malnourished with a human. Same basic concept. Now, there are also stages of fermentation, and we're going to be going through these a lot more over the next two lectures, but I want to point out just kind of the standard sets here. So almost every single fermentation starts off what we call a lag phase or a dormancy phase, where the yeast cells are just sensing their environment. They then go through an acceleration phase. It's not shown here right in this bend up. They then go through a log or exponential phase, a deceleration phase, followed by a stationary phase. We're going to go through this a lot more next week, but this sugar consumption, these nutrient consumptions do follow this path. As the yeast cells grow, the amount of sugars they consume increases as a population. The individual cell stays the same. And the most important thing I want to note here is, much like humans, yeast cells don't like starvation. They can only go so long without food. What happens is, if these yeast cells stay without food for a long period of time, or it can actually even be a short period of time, it's very strain dependent, we go into the death phase. And we start seeing yeast cells start dying en masse. We're going to talk about this a lot more this time next week, and talk about how we can avoid this and what we can do to actually help evade it. And with that, we're going to go on to what makes a yeast cell happy. What are the favorite things of the yeast cell? Oh, yeast cell health, and I can't stress enough, is crucial to make good beer. The happier yeast is, the better your yeast will perform, and the better beer you're going to be producing. Happy yeast, healthy yeast equals no off flavors. This is something that was told to me almost a decade ago, and I've blatantly ripped it off ever since. Is that a better way to think of yourself is not as a brewer, but as a yeast farmer. We can't actually produce ethanol. Yeast produces the ethanol. They're the thing that actually turns the sugary wort that we produce into beer. We're just wort makers. We're in some cases janitors. We're really a yeast farmer. The better you can farm your yeast, the better quality your yeast is, the better quality your beer is going to be off of that. There's no way around that. That is the way it is. Now, if we look at what makes a yeast cell happy, and obviously this is a simplistic view, but we're going to walk through three main bits. Now, consistently fed and stored minimally is the first one, and this is really important. I will often have pro brewers and home brewers ask me, how long can I store my yeast? I have this mason jar full of yeast that's been in my fridge for 3 months, or I have this yeast brink that I pulled off the fermenter 2 weeks ago. How long can I go until it's not okay? I can liken this to you going to a butcher and asking the butcher, hey, I bought the steak from you, or I bought this chicken, or whatever the piece of meat is. How long can I hold on to this thing until it goes bad? It's hard to say. It's nebulous. It has a lot of different variables on this, but overall, the answer is the sooner you use it, the less likely it is to hurt you, and the better it's going to taste for the steak or the chicken, and the better it's going to perform for the yeast. The sooner you can use your yeast cells, the less dormancy they have, the less starvation they've gone through, the healthier it's going to be, and the better your fermentation is going to be. You should always use your culture sooner. I have seen breweries where they do a first in, first out type scenario for yeast cells, and that's not always the best idea. If you know you have, say, a container of yeast or a yeast brink that you don't need, get rid of it and use the fresher stuff. Your beer will improve based on that decision. So that's the first point. Consistently feed it. Don't let it and don't store it for very long. The second one is proper nutrition. Now, if you're making, say, 10-degree Plato worts, and they're 100% all barley produced wort, your nutrition is probably going to be completely fine. It probably is. Now, we have seen internally that a lot of strains have different quirks when it comes to vitamins and nutrients. Some strains are very fan heavy, very fan hungry. Some strains require lots of oxygen. Some strains require certain nutrient profiles that are different from other ones. Adding yeast nutrient or some things like diammonium phosphate is a really good idea. It's kind of like akin to a human taking their vitamins in the morning. It's going to make things go easier. If you are deficient in something, it's going to go away. Yes, you might just be making very expensive urine or moderately expensive urine. But it means that your chance of something going wrong has decreased. Same thing goes with yeast cells, with yeast nutrient or diammonium phosphate. This is going to help things go more smoothly. So making sure the has proper nutrition, kind of feeding it as it needs is important. You have to eat your vegetables. Yeast cells need their vitamins. Kind of the same idea. And finally, and this one can be a little bit more confused. We're going to talk a lot more about this at a later point. Today is the basics. Is proper amounts of oxygen. Like humans, yeast cells really want oxygen. I mentioned this earlier, but aerobic respiration produces 18 times the amount of energy that fermentation does. It is phenomenally more efficient for producing energy. And how the brewers typically handle this is that they'll add oxygen at the beginning of fermentation. They pump the yeast cells full as much oxygen as they possibly can. The reason we're doing this is we want the yeast cells to take that oxygen, go through some little aerobic respiration, produce tons of energy, give themselves this energy boost. It's almost like taking an energy drink or something like that. It's a bad analogy, but you get the idea. Allows them to really get that energy up so they can handle the stresses they're about to go through. Oxygen is also important for other reasons, though. It helps produce some critical compounds that yeast need in order to maintain proper cell health. So if we don't aerate our yeast cells, we don't do that, say, over several generations, we can accumulate some pretty bad problems. Sterols are the main thing that are produced, but we'll talk about those at a later point. Now, another final important thing we really have to stress here is that you should only expose yeast cells to oxygen when they have sugar present. If you've ever bitten into an apple and then you look at where it's been bitten, it very quickly goes brown. Now, the same thing will happen to the yeast cells if they're not currently respiring, if they're not able to use and break down that sugar. The oxygen will go to the outside of the cell and start breaking the outside of the cell, much like we see it turn, oxidize and turn brown the apple. It will oxidize and turn brown the outside of the cell. If the yeast cell can't handle it, it doesn't have sugar to ferment with it, or respire with it, sorry, you should not have it present. You only want yeast cells to have access to oxygen when they have sugar present. There's no other scenario should those two mix. Now, that's kind of the main three. To make yeast cells happy, you want to make sure that you feed your yeast cells frequently, you give them proper nutrients, and you don't put them in a stressful environment. You make sure they have enough oxygen. If you're good to your cells, they're going to be good to you. Do unto others as do unto yourselves. The golden rule. It's important. Now, we're not done with that. We're going to talk about a few other things that we can further do to help yeast cells, but it's really important for us to not just look at what yeast cells like. It's also important for us to look at what yeast cells don't like, because oftentimes a lot of brewers are putting stresses on yeast cells and they aren't aware they're doing so. They're doing things to yeast cells that they think are completely benign or completely fine, and they don't realize that their yeast cells are actually getting really angry. So we're going to go through some of these. So yeast stress, much like humans, is something that typically reduces the ability of them to function. Now, if you're under a very stressed scenario, the chance of you able to respond as you would ideally want to decreases. Now, some stress can be a good thing. Some stress keeps you up. It keeps you going. It keeps you alive with it. Same thing goes for yeast cells. You want the yeast cells to be going through some stress during ferment or during their life. but too much stress can really lead to negative scenarios and can lead to death. Just like for humans, where you see heart attacks and things that increase when you have high stress loads, yeast cell death also occurs due to high stress. So we're going to go over some basic irritants and stressors for yeast cells. Just the basics. We could do a lot more in-depth conversations on this, but we're just going through the basics. So one of the biggest ones is cooling. It's one of these things that brewers don't often think about. Now, when most people think about the ideal environment for yeast cells, and I mean what temperature they enjoy the most, you typically would assume, and because it's how most humans have worked with yeast cells, that right around 20 degrees Celsius, 68 degrees Fahrenheit is where they probably enjoy their life. That's not the case. Yeast cells are actually ideally healthy around 25 to 30 degrees Celsius. And right around 27 C is a pretty good average. That's their ideal temp. So anything we can do to push them higher to that temp is going to make them happier and healthier. And whenever we start pushing them colder, we actually do start stressing them out. Now, the reason for this is we mentioned proteins beforehand. Whenever we chill the yeast cells actually have to produce proteins that help offset that temperature shock, that decrease in temperature. Now, when we warm the yeast cells up, they break down those proteins because they're no longer needed. And they now have all these parts, all these kind of partially built proteins, kind of think like partially built Lego, that they can now go and use and work with. So what we end up seeing, and kind of a result of this, is that whenever we cool the yeast cells, we're actually asking them to expend a lot of resources and energy. Whenever we heat the yeast cells up, we are actually giving them a bit of an energy boost. Now, this is part of the reason why we always recommend, and it's often recommended, it's not just us, that if I want to say ferment a beer at 20 degrees Celsius, I probably want to knock my beer out. I want to cool my beer down 1 to 2 degrees C below where I wanted it to ferment at. If I want to ferment at 20, I want to cool out at 18. If I want to ferment at 23, I want to cool out at 21. I want to give it some space to rise up. That is really, really helpful for the yeast cells. The last thing we want to do is take our yeast cells, which might have been stored for a good period of time, or they could be fresh and very healthy. We don't want to dump them into a stressful environment and then apply a cooling stress to them. It's the last thing we want to do. We always want to make it less stressful as possible. Give them that rise up, make their life easier. They'll thank you for it. Now, one of the other greatest sins when it comes to fermentation temperatures is this. Yeast cells hate this. Now, say we want to ferment the beer at 20. So we set our Inkbird or a Johnson controller, whatever we're using to control the temperature in our facility or in our brewery or at home. We never want to see it go 20, 22, 18, 22, 18. We never want to heating, cooling. What we're essentially doing to the yeast cells is we're asking them to break down proteins, make new proteins, break down new proteins, make new proteins, break proteins, make new proteins. This is a waste of energy and resources that the yeast cell cannot easily get back. And oftentimes this can cause large amounts of yeast cell stress. It's not something you want to have happen. This is part of the reason why you see a lot of brewers try to maintain, say they want to ferment at 20. It'd be 21, 19, 21, 19. It's much healthier. And then near the end of fermentation, just let it free rise all the way up. We'll get into more reasons for fermentation management and things like that as to why that is. Now, to think about the human equivalent to this, it'd be this. It'd be kind of you shivering and then you sweating right after, and then you shivering and then you sweating. That's the human equivalent to what you're asking yeast cells to do if you're going through large temperature swings during the ferment. It's not good for humans. It's not really good for yeast cells either. It stresses them out. Don't do it. Now, one of the big ones for this, and this can be quite impactful, is a lack of nutrients. Now, however, it is very poorly understood, and it's typically really only a thing with high adjunct lager. But we do see this come up and rear its head with a few different other beer styles and things like that, or a few different kind of unique strains. There is a large variance when it comes to required micronutrients for different yeast cells. Different yeast strains will require different levels of certain nutrients. It's highly variable. There is a large amount of research going on this, so hopefully as this comes out, we can all learn more and we'll share it as it comes out. But it's going to be different from strain to strain to strain. Now, to give you an example, though, we go back to our kind of yeast versus human analogy. Now, a yeast with a poor diet, so a yeast cell that doesn't have the proper nutrient components to it, would be kind of equivalent to a human with a bad diet. I've fallen to this before. Eating Little Caesars for many days in a row is not healthy, and it showed. I was not functioning great, poor function. Same thing goes when a yeast cell is not eating what it needs. It's not getting the proper nutrients it requires. What we see is poor performance. For the yeast cell, we see poor fermentations, and this could be materialized as slow or sluggish or off flavors. A whole bunch of different things can arise from this. Or a human can be poor fermenting, not quite mentally clear, poor health, low, shortness of breath, things like that. Not things you want to be under. So one of the other big stressors is too much work. Each yeast cell is only capable of performing so much fermentation, and fermentation is work. It is stress. And it's really important that we all understand that. Fermentation is stressful for the yeast cell. They tolerate it. They don't like it. If we underpitch or we're fermenting a very high OG beer, the human equivalent to this would be essentially if we have less workers. So if you're on a job site or working in a facility and 4 people are sick, each person who is currently there is doing more work. It's just the nature of the beast. Same thing goes for yeast. If I only pitch half the amount of cells I should have, I am asking each cell to go through twice the amount of work that they would have normally. And conversely, they're going to be more stressed. Not conversely, but you know what I mean. Now, conversely, if we overpitch or if we have a very low OG wort, that we have a low workload, that's equivalent to us having more workers. Therefore, every single person has to do less work. We're going home early. It's great, less stress. Now, this is great when it comes to yeast cells as long as proper nutrition is maintained. I've often talked about this as if it's, I have enough food to feed 8 people and 16 people showed up. Everyone's leaving hungry. That's not great. Same thing can happen with yeast. If I only have enough food for 1 trillion of them and I pitch 4 trillion in, each cell is only consuming enough, a quarter of the amount of sugar it should, which is stressful. Every single cell is now leaving, malnourished or improperly fed. Its nutrition has suffered due to this. The balance here is key. So equivalent to this, yeast cells consuming a lot of sugar or a high specific gravity wort, high degree Plato wort, would be like a human having a very large workload. Now, we're going to see poor performance in both cases for this. You can only push yourself so far. The result would be for the yeast cells, a poor fermentation, or for the human, poor functionality, whatever it is, the field that you're currently in. Now, there can also be a handful of other stressors that can cause stress. And now, these are often when we see the presence of antifungal compounds present. And there might be a few of these that are actually you may not have thought of. IBUs, I summarized alpha acids inside of the beer in the international bittering units. They're an antifungal substance or an antimicrobial substance. They are stressful to yeast cells. Yeast cells don't like them. It's part of the reason why we don't harvest yeast cells from dry hop beers. Alcohol on itself is a toxic molecule. It is stressful for the yeast cells. We don't want our yeast cells in its presence for that long. A common one we see, especially kind of this time of year, is caffeine. Caffeine is an antifungal compound. It's also an anti-insect compound. It kills a lot of molecules. If we try to ferment a beer that contains caffeine, those yeast cells are going to be stressed out. And finally, capsaicin, the thing that makes spicy food spicy food hot. That is also an antifungal compound. So if we try to ferment anything with these compounds present, those yeast cells are going to stress right out. And there's a whole bunch more associated with these things. It's not just these 4. There's many, many more. Now, the result of all this will essentially be, if we push this over and over, and will be dead cells. If we don't keep our cells in good health, we're going to have a lot of problems in the brewery. And this poor health, this poor functionality will result in a few different ways. We've probably all been here. I've been here a handful of times. But things like under-attenuation, off-flavor production, inconsistent and uncontrollable fermentation flavors. And this time it tastes great. Next time it tasted very boring or worse, bad. Less generations of the yeast, which means your yeast costs have increased. And increased fermentation time, which means I can't push as much beer through my brewery. All of these are things that can start materializing if we are being hard on our yeast cells. All of these, if we could undo or we could remove, all of our beers would increase. So understand how the yeast cell functions is imperative. We want our yeast cells to be happy. So a poor health yeast cell or low viability would be equivalent to a sick human. Both of these are going to perform poorly. We're going to see poor fermentations, as we just talked about. The human's going to have more errors, more mistakes. It's not going to function as you'd expect or as you'd intend it to be. No one likes this. So to offset this or kind of counterpoint this, we're going to talk about what makes a healthy cell healthy, because this is important. we talked about what can stress a yeast cell out, but we should understand what a good cell looks like. So a good cell, if done properly, it's always done properly. Believe me, we've learned how to not grow yeast many times. They contain a high amount of nutrient reserves. So all these nutrients we talked about so far, they have large stockpiles of things. And this means that you can be a bit more rough on them. You can malnourish them a little bit. You can stress them out a little bit. And they'll be pretty good. They'll be fine within reason. You can do it, say, one plus times, and they'll start showing signs of it. But they do have a much higher level of these nutrients than you'd see if you harvest them off a beer. They've also haven't been damaged. They haven't been going to that high ethanol stress environment. They haven't had to go through, sit inside of ethanol for 21 days or so. They haven't had that stress or damage occur to them. They're fresh. They're primed. They're also fully functional. They don't have any high levels of bud scars that we'll see inside of yeast cells from a prior fermentation. And they have a high amount of energy reserve. We'll talk about this next week, but glycogen essentially is equivalent to the human's fat. It allows yeast cells to survive, and it's what they consume when they're not actively fermenting or actively respiring. Healthy yeast cells have all these things. So if we're able to maintain these elements to them and we're able to remove as much stress as possible, we're going to have very happy, healthy fermentations that are fast, taste great, all the good things we want. So the human equivalent of this is, fresh yeast cells would be a great fermentation. A very healthy human would be Arnold Schwarzenegger from the 1960s and 70s, Mr. Universe, which would be less errors, great health, maybe a movie career, and maybe profit. It's pretty good. We all want to be that. So with that, we're going to kind of finish off on all the different yeast cells we see kind of commonly found in breweries and kind of how they all came to be here. And well, this will really kind of be the launching point for the next couple talks we have. There's different types of yeast we see. Now, all the yeasts you see on this on what we have here are all Saccharomyces cells. They're all common brewery strains. So we have clean ale strains. These are the flavor profile of these effectively is just clean. These would be things like your Cali ale, is what we call it. USO5, WP001, the kind of standard clean ale strain. Nothing fancy, just make clean beer. We have English ale strains, which are almost always fruity and estery, Foggy London, Vermont ale, things like that. Those kind of big NEPA strains, or even more classic like the Fuller's English strains and things like that. They're all fruity, estery, they're light, they're wonderful. Lager yeasts are often drier and cold ferment, and they're crisper. We'll talk about these a few weeks from now, and actually during our webinar conference we do midway through next month. Belgian ales are almost always phenolic, spicy, and they'll often have characteristics of dried fruit. German wheat ales are spicy and banana, clove. Belgian wits are fruity and spicy. Saisons are dry, spicy, and estery. All these different flavor compounds we see above here. The large, vast array of flavor variations and kind of even cell structures seen under the microscope, they're all yeast. They all look very similar. They all handle similar. Almost all the things we talked about today all apply to these. Even though they have massive flavor variations, they are all effectively the same organism. They all work the same way. If you treat all these organisms properly, they're going to produce good beer for you. Now, you may ask yourself, the first time I saw this, I was asking myself this, so I think it's a fair statement, is how we got here. And I want to kind of turn our guys here a little bit. I've talked about dogs a handful of times because I think it's a good analogy for how we got here. And we all know what a dog is. I hope we all know what a dog is. Almost all dogs originate what we believe is the gray wolf. And then through different artificial selections via humans, we started seeing these different subspecies or different classifications of dog arise. So we see things like Shiba Inus and Mastiffs and all sorts of different types of dogs, which then when you then take them from Europe, North America, China, India, and you further break them down even further, we start getting the Cocker Spaniels or the Golden Retrievers or the Mastiffs that we have today. They all originated from the same thing, but by human intervention, we've moved them this way, accelerated the genes this way. Same things happens with yeast. We all took, and all the yeast cells originate with some sort of, proto yeast. But then based on, we wanted this beer a bit more spicy. We wanted this beer a bit cleaner. We want it more fruity. Humans have pushed yeast cells and all these different groupings that we just saw into what they are. These aren't naturally occurring. All the different subspecies of yeast that we just saw are all like the individual subspecies we see here of dogs. Humans have done this. This was by, there was almost by design. Humans have had a huge impact on this, but they all still operate on a basic structure like the gray wolf. It's kind of just a cool analogy for it. Now, finally, this is where we're going to leave on, actually, is this chart right here. And for those who haven't seen this, this is actually the kind of the evolutionary chart of yeast cells. It came out a handful of years ago, but what you're seeing here is the evolution of it, of how all the different yeast cells we use inside the world came to be. So in these, this is not just beer yeast. We see here we have beer 1 and beer 2. But we also have wine yeasts, mead yeast, sake yeast, mixed fermentations, all sorts of different yeast cells that all originate from some sort of common ancestor that's located in the middle of the screen. We will probably never have this yeast, but we do have the impact of all of it. And all of these organisms effectively work exactly the same way that we talked about today. They're all, they all have the same functions. They all act the same way. If you want to get the best key things from them, you better treat them right. And that's it for today. Again, the goal of today is just kind of instill the basics of what yeast fermentation, sorry, not that's the yeast fermentation, but what yeast is. So we can have a better idea as to what yeast does. Yeah, thanks.
Thanks, Nate, for the informative talk. We do have a little bit of time for questions. Before we launch into the questions, I just wanted to mention that we will be hosting the next installment of Yeast Basics this Thursday, March 26th at 8:30 p.m. So same time, just a few days from now. And we'll be talking about yeast flavor production. We'll also be scheduling the next batch of webinars shortly. We'll be presenting webinars every Monday and Thursday until the week of April 20th, where we'll be presenting an intensive webinar series from April 20th to 22nd. We've got some really exciting guests lined up for these webinars, so please stay tuned for more information. So we do have a few minutes for questions here, Nate, if you wanted to get into them. There's a few here that I think may need to be saved for our Q&A period at the end.
I'm going to point this out. I did not drop a Talking Heads reference. I do not know. I don't listen to them.
Okay. Maybe it's just a subconscious thing. The top question right now is actually maybe the hardest one to answer. And the question was, how and why does zinc affect flocculation?
Oh, Jesus.
Yeah, that's one that I didn't know off the top of my head. I started doing some searching. I've got some ideas. The last research I could find on this was from 1990, but I think in our Q&A we can get into some details there.
Yeah, I would point this out though, in that there's a really important thing to mention with zinc, and that is that zinc isn't. Okay, there's two parts for it. You need bioavailable zinc. So not all zinc you add into a beer is bioavailable. So like if you add salt to a beer, that doesn't mean it's fully dissolved and able for everything to work with. It's not the same thing. So that's a really important bit. So just because you're adding zinc doesn't mean it's actually useful to the yeast cells. If you're looking at flocculation, I would, if you're having problems with flocculation, I would probably first recommend you look at calcium, because calcium has been shown to have a very large impact on flocculation. Yes, I've heard bits that zinc is more so. My understanding is zinc is more so associated with general cell health, which could then downstream into flocculation. But calcium really does have a big impact when it comes to flocculation. If you're trying to push flocculation with zinc, I think I would steer you towards calcium instead.
Yep, there seems to be a little bit of research on zinc and flocculation, but it seems to be a little unclear about the role it plays. Just doing a couple little Google searches, I came across some interesting stuff that I think we can string together, and I've actually got an idea to do a little bit of genetic digging, and maybe we can come up with some interesting ideas about what zinc might be doing with flocculation, because there really hasn't been any research for 30 years.
Oh, that 1990 really is 30 years ago. I'm not comfortable with that.
The next question we had was why does the morphology and metabolism of Brett vary so much between strain to strain?
That's a you question, Richard.
I know. One thing that I'll just add for that is that we're aiming to have a whole webinar on Brett. We're going to try to bring in another expert on Brett and try to get really into the details of that yeast, because it's a really special one, and I think it's worth spending some time talking about Brett.
Yep.
The next question we have is, what's the deal with yeast and pressure? I think that's a good question.
In what way?
Well, I mean, I guess the question is, what does pressure do to yeast?
Okay.
Good and bad.
Yeah. So it depends on the type of pressure you're looking at. So if we're talking about hydrostatic pressure, just the weight of the liquid on top of the yeast cell. So if I have, say, a fermenter that is 4 stories tall, we're looking at a high amount of pressure at the bottom of that yeast cake on the bottom. The yeast cells are going to be under a significant amount of pressure, but physical pressure doesn't seem to stress them at a huge amount. What was it? That was from Chris. It doesn't seem to stress them at a huge amount because they can adapt to it. So it's not like humans where if we're going scuba diving, we go up too fast, too quick, we get the bends and our body has to adapt. Yeast cells can just produce the proteins associated with it and react quite quickly. They don't need the time to the same degree we do to adapt to it. What you do see when it comes to a flavor compound standpoint, we'll talk about this over the next two webinars, is that fermenting under pressure does tend to slow the metabolism down of the yeast cell. Spunding valves and things like that are one of the main ways in which this is induced. And by slowing the metabolism, and I'm not trying to bury the lead here, but we're going to talk a lot on Thursday, and this time next week. But whenever you slow the fermentation rate, and you can also do this by cooling the fermentation down, you see fermentation become cleaner because all the accidents, the errors, the side paths reduced in activity. So you see less fusels, less VDKs, less esters, things like that. Now, from a commercial standpoint, you actually see this occur when you see people try to take something from, say, a small, which might be small, like either a 20-liter homebrew batch or like a 5 to 10 hec batch commercially. And I want to scale it up and make 200 hex of it in a giant several story tall tank. Those pressures do need to be offset. So if, say, I fermented the small 5 hec batch at 18, I probably want to ferment the larger batch at about 20 to 21 because I've added a pressure by liquid height, and so I need to remove a pressure by adding stress. Sorry, by removing stress, which one of the best ways to do that is increase fermentation time. So it can be a balancing act between those two.
Great.
Yeah, I think
so.
Yep.
I think so. We've got one question that's rapidly rose near the top. The question is, how do I reuse yeast after a home brew with sediment and yeast at the bottom of the carboy?
We are going to talk about that a lot a week from now. But the easiest way for it is you want to use it as soon as possible. You can take, say, 1 liter or a couple liters of sterile water, add it to your yeast cake, spin it all around, add it to a sterilized mason jar, let it sit and separate. And then if it's less than, say, a week old or so, you can add it right back into the next batch. If you are, we say you want to sit in this yeast culture for a few weeks or even worse, I would heavily recommend you do a yeast starter on that. You want to get the yeast cells kind of going and ripping and things like that. We'll talk a lot more about the details about that on Thursday and this time next week.
Cool. I think we've got maybe time for 1 or 2 questions.
Yeah, sure.
The next one, and I think this might be one we can get into more details in the Q&A, but the question was, what are some good reference materials and textbooks for yeast microbiology?
Okay. The initial one I always recommend is the Jamil Zainasheff, Chris White yeast. That book is by far the most approachable, easy to work with, easy to digest source on yeast. Now, there are a few things that I would, it is, I'd say 95% correct. And for initial running through, it is phenomenal. I recommend that as a textbook for almost all my courses at Niagara College, just because it is such a good kind of basic text. Now, there's some nitpicky areas to it, but that doesn't mean it's not useful or not incredibly useful. If you then want to go to that next step for it, I'm forgetting the authors for it, but it's just called Yeast Fermentation. Bolton and Quain. Sorry? Bolton and Quain.
Yep.
Yeah, if you really want to start getting into yeast cell metabolism, that is by far the book. And the last thing I would recommend, and this is where I do find some people get lost on this, and it might sound a little reductive in a sense. you need practical experience on this stuff. Like a lot of the researchers get kind of in this bubble of under these perfect conditions, under a vacuum, yeast did this. That's great, but that's not what I'm doing commercially. And there is a lot of, unfortunately, institutionalized knowledge for certain strains or for certain, types of beer making that, we're now able to see some kind of like almost like beer archaeology or beer espionage in a sense being done to help elucidate what these things are. But for the most part, not for the most part, but there are a large amount of styles, especially for lagers, that there just isn't the knowledge base, at least in North America, for. If you can speak German, I'm blanking on the name of the book. It's called, not The Practical Brewer, Kunz is the name of the author. And it's this big German, like, brewery text, like Tum in a sense. And if you can read that, that has so much knowledge in it. Unfortunately, I can't speak German and it's not translated into English. To my knowledge, if it is, let me know and I'm buying a copy.
Of Kunz?
Yeah.
I think there is an English translation.
Okay.
I'm not sure how I think you have to buy it directly from the VLB.
Okay.
But I think there is an English translation available.
Okay.
That's I mean, if you want to learn all of these crazy lager secrets, that's the one for sure.
Yeah, and some of those things are just like they're institutionalized knowledge. Like there is that level of we do this because it works versus we do this because we understand why it works. There's a balancing those two is kind of the name of the game in many cases. Mm-hmm. Dan has it. Wonderful. Good to know. $3,000 or $300.
Yeah, it ain't cheap.
Yeah,
it ain't cheap to make lager.
Hi, Amy. I didn't know you were in there.
All right, we're just past the 1 hour mark. We are planning on cutting these things off around the 1 hour mark, just to sort of keep the interest of time and keep the pace going and save some questions for our upcoming Q&A session, as well as some of the future discussions. So I think we're going to cut this one off shortly. Although I do appreciate everyone coming in here and chatting. What was that, Nate?
Based on the chat, apparently we need to talk about tofu.
Tofu. Okay. Interesting. Maybe that's a future topic.
There you go.
Uh-huh. Great. Okay. Well, thank you everyone for tuning in, and especially for the people who have been active in the comments section and asking questions, and hoping to see you on Thursday, where we're going to get into yeast flavor production and start to dive in and get a little bit nerdier. Cheers.
Yeah. Cheers.